Well, welcome back to the Bio180 lecture series. Today we're going to be continuing our exploration of cell metabolism. And so again, just a map of where we've been and where we're going. We have already completed the first 3 lessons of what we called our biochemistry boot camp. And so those are done. And in our last lecture, we started looking at glycolysis as the first step in our process called cell respiration. So we talked about glycolysis, and now today we're going to be continuing or extending glycolysis and talking about the next step in that pathway, the citric acid cycle. In our next lesson, when we come back, we'll continue from citric acid cycle to the electron transport chain and ATP synthesis. And then finally, these last 2 days we'll spend talking about photosynthesis as a catabolic pathway. But again, today we're going to be talking about the citric acid cycle. So we'll briefly review where the citric acid cycle fits in the cell respiration pathway. Then we want to go through the pyruvate transport, or the bridge between glycolysis and the citric acid cycle. We'll look at the citric acid cycle itself. And then citric acid cycle is going to, well, we came through glycolysis into the citric acid cycle in order to break down sugars like glucose, but we also eat and therefore break down other things for energy as well. And so we're going to see how we use a process called beta oxidation to break down fats, and a process called deamination to break down amino acids so that we can eat fats and proteins and digest them for energy as well. So let's come over here. And we had previously looked at glycolysis as the start of our cell respiration pathway. And we said that going into glycolysis, we had a glucose molecule, and then coming out of glycolysis, we had pyruvate. And so pyruvate was this 3-carbon molecule. So it had 3 carbons in it. And then we said pyruvate could go one of 2 routes. That if we had oxygen, if we had no oxygen, so no O2. By the way, we would call that anaerobic conditions. Anaerobic. Or you'll often, or sometimes, maybe not often, hear it called hypoxic conditions or low oxygen conditions. And if we did that, then we went into some of our fermentation pathways. So we had alcohol fermentation, or we had lactate fermentation, and we discussed that. But today we really want to continue on if we do have oxygen. So if we do have O2 up here, this is called the aerobic pathways. Then we're going to do something different. And that's where we want to start our pathway right now, is we're going to take pyruvate. We're not going to see the oxygen yet. That's actually not going to come until our next lecture series. But if we do have oxygen present, we're going to go in this pathway. And so pyruvate, let's extend this arrow out a little bit. Pyruvate is going to be converted into a molecule called Succinyl Coa. And this step right here, this is called pyruvate transport, or sometimes just a pyruvate bridge. And so that's the process we're talking about. And the reason why it's called pyruvate transport is because glycolysis over here is happening in the cytosol of the cell, and that's where we have pyruvate, that's where we create it. And then over here, when we get to the citric acid cycle over here, this is going to be in the mitochondria. So we'll put up here mitochondria. So that's the location of that. So somewhere in this area, we've got to go from the cytosol to acetyl-CoA actually will be in the mitochondria. And so that's why we're transporting it. We're going to transport it across the mitochondria membrane. And actually we should put membranes, plural, because we're going to transport that across both the inner and the outer membrane. Turns out that really we're going to transport it across the inner membrane and put it into the mitochondrial matrix. And that's where all of this process of the citric acid cycle is going to take place. In the process, notice we're going from a 3-carbon. Acetyl-CoA actually has only 2 carbons. So somewhere we lost a carbon, and there's actually a lot going on in this particular reaction that we want to look at. So one of the first things that happens, well, maybe not the first thing that happens in the reaction, but notice we went from 3 carbons to 2 carbons. So we lost a carbon somewhere. So we lost it. I'm going to draw an arrow coming off here. So one of the things that happens is we lost a CO2. And over here, let's draw the structure real quick. So this is what pyruvate looks like. So this is pyruvate. And what happened in this case is we had an enzyme, a coenzyme. We had a molecule called Coenzyme A. We abbreviate that and we just call it CoA. And CoA, I'm going to draw on here, has a sulfhydryl group. So this is one of the functional groups that we learned earlier in the classroom. And coenzyme A acts as a coenzyme, and it has this sulfhydryl group. And what happens is we learn that this, well, either one of these can be a good target, and CoA actually attacks right there. And it forms a bond with this carbon. But now, as we've learned in previous lectures, this carbon has too many bonds to it, so it has to get rid of one. So it gets rid of that bond right there. And that bond, those electrons, are going to reform over here. We get rid of that negative charge, and that becomes our carbon dioxide. So what we're really going to do is take a carboxyl group, and we are going to oxidize it. So we have an oxidation. Right here, sulfur has too many bonds. We're going to lose that bond right there. And that hydrogen, with its 2 electrons, has to go somewhere. And that is going to be our reduction step. Remember last time or a couple lessons ago, we learned that anytime you have an oxidation, you must have a reduction. And so we have our CO2 formation. That was our oxidation. And then we have this hydrogen is going to go somewhere, and that's going to be our reduction. So where does it go? Well, in glycolysis, we talked about anytime you need a reduction, we are going to pull out a molecule of NAD+, and that is going to receive that hydrogen ion right there, anion, and it becomes NADH. And so that's where the hydrogen went. So in this step, we are releasing CO2, but we're also creating an NADH, and that's going to be important. Today, we're going to talk a lot about the creation of NADH and FADH2 as these energized coenzyme carriers. We'll also add to this the fact that we did bring one other thing into it, and that was our molecule of coenzyme A. And so we lost the CO2, and that's how we formed acetyl-CoA. Even though I went through some of the structure just so you could see that it's not magic, that there's just actual chemistry coming here, you can follow along with it. This is really what I want you to know about the reaction. Simply that we're going from pyruvate to acetyl-CoA. You need to know those 2 intermediates. And then these steps, just that you have an NAD plus coming in at this step, forming an NADH. You have this oxidation, CO2 is coming off. And then, of course, that we have the CoA coming into the reaction. And that's the pyruvate bridge. So it's just a one-step reaction, but we have a lot going on. We're transporting from the cytosol into the mitochondrial matrix. That's the first thing we have going on. We have some energy production right here that's being stored in that NADH molecule, and we have the release of CO2. Now, the acetyl CoA is then going to enter into the citric acid cycle. So the CoA molecule is actually what we call an energized molecule. We've learned in an earlier lesson that we could take a molecule and we could energize it by adding a phosphate group to it, right? And that would raise the potential energy up. CoA is going to work in much the same fashion. When we add that CoA onto the acetyl molecule or the acetate molecule, it raises up the energy. And this molecule right here has enough energy that it's going to be able to form a new covalent bond. It's going to attack citrate, or sorry, it's going to combine with this molecule called oxaloacetate in order to form a new molecule called citrate. And the key to that is that it is a CoA molecule. If this were just no CoA and we're just an acetate group, it wouldn't have enough energy to do that. That's why we had to add that CoA in the pyruvate transport. It was going to energize that and allow us to make the new bond to form citrate. So so now we have in here, this is the citric acid cycle. And we're just going to walk you through the important elements. Citric acid cycle of this cycle. So in the first step, acetyl-CoA, a 2-carbon intermediate, is going to combine with oxaloacetate. And one of the important things to track in the citric acid cycle is going to be the number of carbons. So we're going to be frequently referring to the number of carbons of each intermediate. These top ones will tell you the number of carbons. We're not going to focus too much on their structures. And for the citric acid in general, citric acid cycle in general, I want you to focus on the number of carbons more than the structure. Although later on I am going to come back and show you some chemistry that's going on with the structures. That'll come up later. But getting back to our citric acid cycle, we're going to take a 2-carbon acetyl-CoA. We're going to combine it with the 4 carbons from oxaloacetate, and that's going to give us the 6-carbon intermediate called citrate. Citrate is going to isomerize, and so we saw this a little bit last time. So this is an isomerase enzyme. The name of the enzyme is isomerase, and it forms isocitrate. Isocitrate, since it's an isomer of citrate, also has 6 carbons. So not a lot going on in that step. Nothing comes in. Well, technically speaking, water comes in and then water goes out. It's a zero net sum, so we don't really worry about that. But we have isocitrate. The next step, however, does get exciting for us. So in the next step, we're going to take isocitrate with 6 carbons, and we're going to convert it into a molecule called alpha-ketoglutarate. And alpha-ketoglutarate has 5 carbons. So again, like we learned with glycolysis, we go from 6 carbons to 5 carbons. We ask ourselves, what happened here? And the answer is the same thing that happened before with pyruvate. We lost a CO2. In a way that's similar to our loss of carbon with pyruvate, in that process, we also took another NAD+, and we converted it into an NADH. In fact, everywhere in pyruvate transport and the citric acid cycle, when we lose a CO2, it is going to come with the formation of an NADH, because every time we lose this CO2, it's an oxidation, and therefore we have to have a reduction where those electrons can go. So loss of CO2 automatically is going to cause me to write down a formation of NADH as well. Here's our oxidation. There's our reduction. In the next step, we're going to have alpha-ketoglutarate form into a molecule called Succinyl Coa. Now, if this looks a little bit familiar to you, good. Notice that we have this CoA, this coenzyme A, being used again. That implies, hopefully you'll think, oh, that looks kind of like my acetyl-CoA formation. And if you're thinking that, then you would be right. The enzymes that carry this out are very similar to each other. They're close cousins. They have the same reaction mechanism. So we are pulling a CoA into the reaction. Succinyl Coa itself has 4 carbons, 4-carbon intermediate. Hopefully you're saying, oh, 5 carbons to 4 carbons. I lost a CO2. And you would be correct. We lose a CO2 at that step. And because we lost CO2, that was our oxidation step. We oxidized the CO2. We need a reduction. Where is that happening? Well, we're going to take another NAD+, and we're going to create, yet again, another NADH. Now, at this point in the pathway, these steps here on the top of the board are referred to as the CO2 emission stages. So we actually divide the citric acid cycle in 2 different stages. This top part is called the CO2 emission stages, and you can see why, because these are the stages that we've emitted our carbon dioxide. And we're actually all done with emitting carbon dioxide now. Let me emphasize one point to you. As we come back and look over here, we had pyruvate that had 3 carbons on it. Well, we lost 1 carbon, so now we had 2 carbons left of that pyruvate. As we went through this pathway, here we lose a 2nd carbon, so now we have only 1 carbon left of the original pyruvate. And then here we lose another carbon. So that by the time we get to Succinyl Coa, really all of the carbon that we had in our pyruvate is gone. We have literally just taken that pyruvate that we got from eating food. Remember, it was a breakdown product of glucose. So when I eat glucose, it breaks down into pyruvate, and then that pyruvate, through the CO2 emission stages, is actually converting into carbon dioxide. So I have actually lost all of that pyruvate. And this is what happens to the food we eat. When you eat food and you process it metabolically, you end up breathing it off. And that's where it goes to. I always think that's kind of interesting that you breathe off your food. So I always tell students, if you want to lose weight, breathe harder. And that will get rid of that extra food that you're eating. To breathe harder, you might have to do exercise or something, but that's the key, is to breathe harder. So, we're now at Succinyl Coa. This next step, something important is going to happen. Remember, we said that when you have CoA attached to you, you're energized. You have a higher energy level, just like as if you added to a phosphate. And in fact, that's going to be kind of what happens. We're going to bring a phosphate. I'm just going to put a P with a circle in there. We're going to bring a phosphate into this reaction. It ends up replacing this CoA to form a molecule called Succinyl Coa. And that phosphate, we're also going to bring in a molecule of ADP. And we are going to create that phosphate in that ATP is going to use the energy in Succinyl Coa to create a molecule of ATP. And remember, in cell respiration, this is ultimately what we're trying to get is ATP. I will tell you, I'm going to make this a little bit fuzzy because I'm not telling you the whole picture. What we actually do is we bring in a molecule. The steps of this, the way the enzyme works, is actually to bring in a molecule of GDP, and then we form GTP first, and then the GTP creates the ATP. So this is an intermediate. We don't really see this, but that's actually what's happening there. If you get into a biochemistry class, they'll talk more about this intermediate step. But here's what I want you to know. The bottom line of this is Succinyl Coa takes a phosphate, ADP, we make an ATP at this step, and then we have the succinate molecule that's formed. I guess we ought to put that we also have, we lose our CoA in the process, right? So that comes off as a product and goes back up here and can be reused. Bottom line, in this step between the two phases, we are actually making an ATP from the conversion of Succinyl Coa into succinate. We now have succinate. It's a 4-carbon intermediate. We've at this point lost all the carbons from it that pyruvate had given to it. And so we're now going to go into what we call the bottom half of the cycle. And this is called oxaloacetate, or just OA regeneration. The whole purpose of this bottom half of the board is to take succinate and convert it back into oxaloacetate so that we can run the cycle again. That's why it's called the citric acid cycle. It has to be able to regenerate itself so that we can keep running more pyruvates, more acetyl-CoAs through. So how do we do this? This is where the structures are going to come in, and hopefully you'll be able to see these on the screen. I've drawn the structure of succinate down here on the bottom corner of the board. Let me just point out a couple features. We have succinate itself isn't too complicated. It's got 2 carboxyl groups on either end of it. We're not going to change those much as we go through. All the business is going to happen in these steps right here, the 2 middle carbons in there. And you'll notice that they have, this carbon has a maximum number of hydrogens on it. So we would say that this carbon is actually fully reduced. And now when we come over here and look at oxaloacetate, notice that this carbon is still fully reduced. It has its hydrogens on it. But this carbon right here has changed. It's gained bonds to oxygen. In other words, it's been oxidized. And that's going to be a key to this bottom half. We're looking at the oxidation, several oxidation steps that are going to happen. So let's go through that a little bit, because it also tells us what happens in these steps. From succinate, one of the first things that happens is we are going to take this hydrogen, and we're going to pull them off of succinate. And when we do that, notice that over here, our next intermediate fumarate is missing those hydrogens. The electrons that were in that bond, we're going to do a couple things with them. 2 of the electrons are going to go to form a double bond right here. Even though we're not with oxygen, because we removed electrons from it, this is an oxidation step. Anytime we have an oxidation, we need a reduction. And so we're going to take a molecule called FAD, and we're going to take these 2 hydrogens, and we're going to add them to FAD to make FADH2. And that's going to be our reduction step right there. So we have an oxidation from succinate to fumarate, and we have a reduction from FAD to FADH2. Now, FAD is a new coenzyme for us. Previously in glycolysis, and we've been seeing up here, we had this NAD plus coenzyme that we're reducing. This is just a new one. FAD has to take place in this step because there's not quite as much reducing power in this reaction. And so there's not enough power to create an NADH. And so we have to use an FAD to make an FADH2. It requires less energy to do that. It's kind of like the difference, I'll tell students, NADH is kind of like a AA battery. It's bigger, it has more power. And FADH2 is kind of like a AAA battery. It still holds energy, but not quite as much. In our next step, we've got fumarate. We've got this double bond right here. And we are going to add some water onto it. And this is kind of, we can think of this a couple different ways. It is going to be an oxidation step because we're going to, in malate, right here, we're going to add a bond to oxygen on that carbon. So it's an oxidation. But it's also the water, what's going to happen is it's going to add right across that double bond. So we're going to break this double bond and reform it with those two. The water ends up being reduced, or this carbon becomes reduced, and then this carbon is going to become oxidized. So this is kind of an internal oxidation reduction reaction. We don't need any NAD plus or FAD in this step. But we do form malate. One of the ways that a lot of students have learned this or remembered this step in the past, the fact that we have water coming in, they use the name fumarate. They say fumarate is fuming hot, so we give it some water to cool it off, and then we form malate. Malate has this hydroxyl group up here and a hydrogen, and in our last step, we are going to remove this hydrogen right here with its 2 electrons, and we're going to take this one off, and we're going to form a carbonyl group. We're going to reform that bond, and then we'll form oxaloacetate. So now we've got 2 bonds to oxygen. This is then an oxidation step. So we need a companion reduction step. This one does have enough power. That we're going to form an NADH out of it from an NAD+. So, as we step back and we look at the board, this is, now we've regenerated oxaloacetate and the cycle's done. So that's the citric acid cycle. Again, just to review, what do you need to know? Anything that's written up here in blue, so the steps coming in and out, just like glycolysis when we learned it, you will need to be able to know and track the steps coming in and out. The stuff in black, the actual intermediates in this pathway, you don't have to memorize because we're going to give them to you in that cheat sheet that's going to be on the test, or you can look them up in the Google or the internet if you ever need them. So you don't need to know anything in black. You don't really need to know the structures either. I just wanted to explain down here because they're all 4-carbon intermediates, what it was that was changing or different between the intermediates. As far as enzymes go, in green, isomerase right here, that's a pretty easy one to remember because it's creating isocitrate. Anywhere that we are going to see the NADH formation or FADH2, that is going to be a dehydrogenase enzyme. So that's a dehydrogenase. This is a dehydrogenase. This is a dehydrogenase. And this is a dehydrogenase. And those are the enzymes that we really want you to know about. And then again, everything in the blue. Purple, know that you have 2 different stages, the CO2 emission stage and the oxaloacetate regeneration stage. So that's what we want you to know about the citric acid cycle. Let's do a little accounting and a summary of pyruvate transport and our citric acid cycle. So we started out with pyruvate. So for the pyruvate transport, we started out with pyruvate. And out of that, we got an Acetyl CoA. And we're going to put here, we also had an NAD plus that we put in, and we got an NADH, and we got a CO2. We also, I suppose we could put up here that we got a CoA. And then this was Pyruvate Transport. And really, we had one of each of these per every pyruvate transport. We then did the citric acid cycle. All right, citric acid cycle. And so in the citric acid cycle, we put in 1 acetyl CoA, and then out of that, we actually ended up pulling off 2 CO2s. One acetyl CoA. And I suppose we also got a CoA off. We had, what else did we have over there? We had some NADHs that were formed. If we go over to that side, in fact, let's come over here. So let's look at our NADHs. We got 1 NADH right there per cycle. Here's a second one. And then here's a third one. So we actually got 3 NADHs. So we'll put that down. And to get those, we needed some NAD pluses. We created an FAD, so we put in 1 FAD, and we got out 1 FADH2. And then, importantly, we put in 1 ADP plus an inorganic phosphate, and we got ATP out the other side. And so there was 1 ATP, and then we put in 1 of each of those. We also had a couple CoAs come in here and out. They're going to cross-cancel out for us. In the end, notice that from the citric acid cycle, we did get 1 ATP, but not a lot of ATP out of it. If we go back and look at glycolysis, we got 2 ATPs out of glycolysis. This would be 1 time through the citric acid cycle. Remember, we can run from glycolysis 2 pyruvates through this thing. So we could double all these if we're looking at 1 glucose molecule. But still not a lot of energy out of ATP so far. Our pyruvate is all gone. Earlier we said that there was a lot of energy locked up in that pyruvate and that we were going to tap into it and pull it out in the later cycles. And so we're in a later cycle now. The pyruvate's all gone. But we still don't have a lot of ATP to show for it. Where did all our energy go? Our energy is locked in these molecules, in these, we call these reduced coenzymes. And so we created 1 of them up here. We created 4 of them right there, and that's where all our energy is locked into. And so later on in our next lecture, we're going to be looking at this NADH2 and FADH2, and we're going to go forward and carry them into the electron transport system and see how we unlock the energy that's held in the bonds of those reduced coenzymes. And so that will be our next lecture. But for right now, we did get some ATP for efforts. This is where the majority of the energy is. And this is a summary of where we've sort of been with the citric acid cycle. Now I want to come over here. So we've talked about pyruvate transport. We've talked about the citric acid cycle and done a summary. So far, if we use glycolysis, we're doing sugars. We're now going to look at these two processes, beta oxidation and deamination, and see how these processes are used to generate energy out of fats that we eat and proteins that we eat. So now let's start first with this process of beta oxidation and how we use it to break down fats and get ATP out of it. So if we come over here, I've drawn out roughly the structure of a fat. Here's my glycerol backbone that we've learned about previously. And then I've got 3 acyl groups, or fatty acids, that are attached by my ester linkages. And the first thing that's going to happen is we're going to get hydrolysis of one of these fatty acids. So we're going to have coenzyme A that we learned about earlier is a good attacker. And so coenzyme A is going to come in and attack one of those ester groups. We're going to break that bond, and we form a fatty acyl CoA is what it's called, a fatty acid CoA. And so that's the first step to breaking down fats, is we create the fatty acid CoAs. Now, over here, I've drawn not the complete structure, but at least a partial structure of a fatty acid. Here's my CoA that attacked it. And this is the carbonyl carbon, or the carboxyl carbon, depending on how you look at it. And then chemists have a notation. This second carbon right there is known as the alpha carbon. And then this third carbon right here is known as the beta carbon. And that's where we get this name, beta oxidation, from. So we want to be watching this carbon really closely. We're going to oxidize it. And so the first step in that oxidation is we are going to create a double bond. We're going to do that by taking off these hydrogens right here. And they're going to each have an electron with them. And then the other 2 electrons that were in these bonds goes back there. And so the first thing we're going to do is form a double bond between those. Now, these 2 hydrogens right there, they can't just hang out with 1 electron. You can't have, they need to fill their octet rule. And so we are going to take those and take a molecule of FAD, and we're going to add those hydrogens onto it to make FADH2. So we're going to add those 2 hydrogens to make FADH2. Now, this was a reduction step. We learned that earlier, so here's my reduction. The oxidation was these 2 carbons got oxidized. There's no oxygen present. This is kind of like, in fact, it's the exact same reaction that we saw taking place over here between succinate and fumarate, where we grabbed 2 hydrogens and pulled them off and stuck them onto FAD to make an FADH2. It's exactly the same chemical reaction. In our next step, we're going to actually take water and we're going to break that bond right there. And we are going to form the hydroxyl group is going to go right there, and then we're going to get a hydrogen added right there. And so that's the next step, is adding water. Okay, so we had step 1, or I guess it'd be step 2, was going to be double bond, FADH2 form. Step 3, add water. We'll erase this now. In step 4, we're going to do what we did before. And in several steps, we're going to take off this hydrogen. We're going to take off this hydrogen with its electrons. And then we're going to reform a bond right there. And so that we get a carbonyl group forming. This hydrogen that we broke off right there with its electrons is going to go and be grabbed by NAD plus to make an NADH. And so that was our fourth step, was we're going to form an NADH. So this was a reduction step, and notice this one we literally added another bond to oxygen, and so that's our oxidation step. In our final step, we are going to add now a second CoA. Remember we said that CoA was a good attacker. We've got a good target right here. And so the CoA comes here. We break this bond, and the CoA reforms that bond. And now what we have, this unit right here is just acetyl CoA. If that sounds familiar to you, it should. This acetyl CoA is going to come back and add right back into the citric acid cycle. So, I don't necessarily want you to remember all these steps. I'm showing you the chemistry of what happens, so it's not magic to you. But ultimately, here's what I do want you to be able to do and understand. For any fatty acid, when you're given a fatty acid, it's going to, through this progressive rounds of beta oxidation, we're going to eventually slice it into 2 carbon intermediates. Before, so we pulled off this CoA, now we have 2 new carbons. We've got a new CoA with a carbonyl group here. The process would repeat on that one, and we'd split. So what happens is we oxidize that carbon, then we go on to the next one, split here. We oxidize that carbon, move down to, oxidize that carbon, move down to, oxidize that carbon, split, and oxidize that carbon. And through progressive rounds of beta oxidation then, we're going to take that fatty acid and we're going to chop it up into these 2 carbon acetyl-CoA intermediates. Every time we do that, and this gets important, every time we oxidize this, we got 1 FADH2, and we got 1 NADH out of it, right? And then same thing here. We got an FADH2 and an NADH every time we carried out that beta oxidation. So, in this case, we had a fatty acid. I made this kind of a shorter one. We had a fatty acid that was 12 carbons long. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And so out of beta oxidation, we got out of it acetyl-CoA. Now, we started out with 12 carbons. How many acetyl-CoAs did we get out of it? Well, we got 1 here, 2, 3, 4, 5, 6. So we got 6 acetyl-CoAs out of it. Great. Those are going to run through the citric acid cycle, and they're going to be able to produce more NADHs, a couple FADH2s, and even some ATPs immediately. But just as important, we got some FADH2s. Remember, these are the high-energy reduced coenzymes that we're going to use later to get even more energy. So how many of those did we get? Well, we got 1, 2, 3, 4, 5. 1 for each beta oxidation we carried out. So we got 5 FADH2s. And then we also got 5 NADHs. So now we've broken down our fat. It'll go through and we'll get even more FADH2s and NADHs out of the citric acid cycle. But just the beta oxidation of that fat really created a lot of reduced coenzymes for us, and these will produce ATP for us later on. So that's pretty significant. This formula, so we started with 12 carbons. Let's go ahead and give you guys a formula. If you take the number of carbons, how many acetyl-CoAs will you get out of a fatty acid? Just count the number of carbons and divide by 2. And that equals the number of acetyl-CoAs. So that's not hard math. If I had an 18-carbon fatty acid, then I get 9 acetyl-CoAs out of it. How many FADH2s do I get? Well, I get C divided by 2 minus 1 equals the number of FADH2s, and that also equals the number of NADHs. Now, why is this less? Why do I get 6 acetyl-CoAs and only 5 NADHs or 5 FADH2s or 5 NADHs? Well, it has to do with the fact that this first carbon right here, the original fatty acid that we broke our first acetyl-CoA off of, it was already fully oxidized, so we didn't do a beta oxidation on it. So we couldn't get any NADHs or FADH2s off of that first one. We only got it off the other carbons in the pathway. So this is how, given any fatty acid, you can tell how many acetyl-CoAs, FADH2s, and NADHs using this formula. One of the things that's interesting is if you look up in the literature, maybe you've heard this before, is that fats will carry a lot more energy in them than either proteins or amino acids that we'll go over, or even sugar. So if you need a lot of energy storage or food that's high, rich in energy, then you really want to carry it in fats. So for example, grizzly bears up in Alaska during the salmon run season, you'll often see them. There's so many salmon coming in, and they're trying to store a lot of fat and a lot of nutrients for the long winter. Sometimes they'll catch the salmon, and what we think of the best part of the salmon is the meat. They don't even go for the meat. They'll catch the salmon, and then they'll just take either the eggs, which are rich in fat, or just the fat that the salmon has. And then they'll throw the rest because they're just trying to get as much energy. They don't want to waste time consuming the meat. They want to get another salmon and eat more fat because it has so much energy in it. Why? The energy is coming from the fact that these carbons in here, remember, were fully reduced. There was no oxygen on them at all. And so by oxidizing them, we were able to get energy from it. Glucose, when you look at glucose, already has all those hydroxyl groups on it. So glucose is already partially oxidized. And so as we go through glycolysis and citric acid cycle, we continue to finish that oxidation, but here we get more oxidation steps. And that's why fats hold more energy. We also see that we have here, this would be a pretty short fatty acid. So one molecule of fatty acid, this one was short, had 12 carbons in it. Glucose only starts with 6 carbons. And so we don't even have as many carbons. So fats provide more carbons for us. And because they have more hydrogens on them, they're more fully reduced to start with, we can do more oxidation steps, and we can get more energy out of them. And that's why fats carry more energy. Now, let's go back and look at our last thing. So we like eating fats, we like eating sugar because they have that energy that we want. Let's go back and look at our last nutrients, which is going to be amino acids or proteins. And how do we digest proteins then and get energy out of them to make ATP? For proteins, we are going to use a process called deamination. Now, that can seem like a big word and it's hard to pronounce, but the key word in here is amine, which is derived from amino. We're going to de-aminoize it. And so we can take a structure. So well, first of all, we're going to start with a protein. And the first thing that we're going to do to that protein is we are going to digest it. So we're going to hydrolyze it into its component amino acids. Let's take as an example one of those amino acids I'm going to draw is going to be glutamate. So glutamate has a structure like this. Has a 5-carbon chain. It has 2 carboxyl groups at the end. And then right here, we have the amino group on it. This is a little bit different than the way I usually draw them. I'm emphasizing the carbon chain right here. So it has an amino group coming off of it. Here's the amino, here's the carboxyl. This would be the side chain of glutamate. Biochemically, there's a couple of ways that happen, but really the second step that we're going to do is remove ammonia from there. So basically what we're going to do is we're just going to break that bond. We're going to deaminate it. We're going to remove ammonia or remove that amino group from that structure. Now, I'm not going to go into how or why this happens. There's a couple different ways of doing it. But when we remove that amino group, it ends up getting replaced by a carbonyl group in there. And so we take the amino group off. Biochemically, there's a couple different pathways that will do this, but we add our carbonyl. Now, the nice thing about this is now we don't have an amino group. That's going to be taking that ammonia that we took off, is going to be put through another biochemical pathway to make urea, and you'll excrete it from the body. But here's the nice thing. We've now got this 5-carbon intermediate, and that is going to go into the citric acid cycle, so we can break it down and get some coenzymes out of it, or some ATP out of it, or actually, and some ATP out of it. To know where it goes into the citric acid cycle, we have to count the number of carbons. So glutamate has 1, 2, 3, 4, 5 carbons, or had 5 carbons. And so when we go over to the citric acid cycle, it's going to enter, in fact, when we remove that amino group, we actually created alpha ketoglutarate. So the structure that I finished off with, this structure is no longer glutamate, it's actually alpha ketoglutarate. And it will enter and run through the citric acid cycle until it's all broken down by losing its CO2s, and that's how we'll get the energy out of it. If you have a 4-carbon intermediate, it's going to tend to enter as oxaloacetate. If you have a 6-carbon intermediate, it goes in as isocitrate or citrate. If you have a 3-carbon intermediate, it actually goes in before, it goes in as pyruvate and runs through pyruvate transport. And then if you have a 2-carbon intermediate, it goes through as Succinyl Coa coming through. And so that's how we take the amino acids and break them down for energy. So, to summarize, we've gone through sort of a brief overview of cell respiration to this point. We went through just glycolysis and how it fit in with pyruvate transport and citric acid cycle. We then went into detail about pyruvate transport and what happened and the citric acid cycle. And that's how we break down sugars. So we use glycolysis and citric acid cycle. And then we saw that we can take fats and break them down through beta oxidation. We can take proteins and break them down through deamination. And all of this, whether it's sugars, carbohydrates, or proteins, or fats, all of them are then eventually funneling into this magnificent hub, this metabolic hub of the citric acid cycle. And in fact, the citric acid cycle is one of the primary central hubs of metabolism in the human cell. Every molecule seems to pass through this cycle at one point or another, and it's key to energy production in the cell. So, with that, we'll end today's discussion and invite you to come back and look at how we're going to take our NADHs and FADH2s that we created in the citric acid cycle and generate the ATP that our cells need to run as we look at the electron transport chain next time.